Hello again, welcome back. For the remainder of this course, we're going to be talking about viruses infecting organisms. And today, I want to talk about some of the basic principles of infection, how viruses get into hosts, how they spread, how they find their target tissues, and how they spread to a new host. So far in this course, we have been talking about viruses infecting cells in a culture dish. And of course, that is not where viruses exist in nature. They infect host organisms, animals and plants and fish, and of course many others. And in an organism, it's a different story. You simply don't add virus to a plate of pure cultured cells and watch replication happen. So these are the challenges that we're going to be talking about for the rest of this course, how viruses get in, the host defenses against them, what happens inside the host, and so forth. In a host population, the viral genome must establish itself to endure. In a culture dish in the lab, it's a one-shot deal. You infect the dish, you harvest it for your experiment, and it's over. But in nature, viruses have to establish themselves, which means they have to find a new host after they're finished replicating in the first one. For both systems, though, a culture dish or an animal, what does hold in common is that viruses have to get in and they have to get out. We'll start talking about that today. As I said on day one, we live and prosper in a cloud of viruses. We are constantly assaulted by viruses in the air, in the food we drink, in the surfaces we touch. But as you know, most infections have no consequence. We talked about some of the reasons for that before, like they just pass through you or they don't cause any pathology. But here today is a new concept. For those viruses that do infect us, which means the viruses get into cells and they replicate, many infections are inapparent. So that's a word I'm going to use a lot, an inapparent infection. Another word for that is an asymptomatic infection. Viruses are replicating in the host, but it doesn't look like they are, not unless you look very closely. But on the surface, there don't seem to be any symptoms whatsoever. Let's look at an example of that. West Nile virus, great example. West Nile virus is a flavivirus, which you have encountered before. These are envelope viruses with plus-strand RNA genomes. West Nile virus is vectored by a mosquito shown here. These are Culex mosquitoes or Culisine mosquitoes. This virus was not present in the Western Hemisphere before 1999. It was in other parts of the world. It came to the U.S. in the summer of 1999. It came right here in New York City for the first time. And some very astute people noticed that that summer, birds were dying, and particularly in the Bronx Zoo, a lot of exotic birds were dying. And it took some very smart veterinary pathologists to figure out that this was no ordinary death, that it was a virus causing this death. And at the same time, people were getting sick in New York City, a number of people developing infections of the brain or encephalitis. So this virus came to New York City in the summer of 1999. It seems to have come from a goose farm in Israel. The virus that arrived in New York is basically identical in sequence to a virus found in a goose on a farm in Israel. The virus came to the U.S. and then slowly spread across the U.S., as you can see by this series of maps on the right of the U.S. And by 2008, it had spread across the country. It is now present throughout North and South America. And it's an interesting story because it had never been here before. Everyone was not immune at all, and therefore the virus spread readily. The spread of this virus was assisted by the fact that mosquitoes not only infect people with these viruses, but also birds. Birds are a huge reservoir of this virus. And other related flaviviruses like dengue and Zika virus, they can be introduced into the U.S. We've seen it happen multiple times in Florida, but they don't spread. And that's in part because of the vector, which is less distributed than Culisine mosquitoes, but also because there is no non-human reservoir. By October of 2004, about 1 million people were infected. We know this because we did serological surveys where we take serum from people and we measure antibodies against the virus. About 20% of infected people developed a fever. About 1% developed neuroinvasive illness. The virus goes into the central nervous system and causes damage there. But 80% of infected people had no obvious disease. This is what we mean by inapparent infections. This is common for many viruses, and it is a problem because it makes it difficult to recognize an epidemic. This is a theme we'll come back to when we talk about this sort of virus infection. The infection of hosts with viruses is used to understand viral disease. Much about what we'll talk about today and for the rest of this course is called viral pathogenesis. Viral pathogenesis is the production of disease in a host infected with a virus. Of course, the virus causes some of the disease, but the host is equally guilty, perhaps even more. The immune response we make to a virus infection is often the only reason why we feel badly. So, for example, when you get influenza and you have runny nose, sniffles, sneeze, sore throat, fever, aches, most of those are because of your immune response and not the virus itself. There are certainly viruses that cause tissue damage, and that's part of the pathogenesis, like Ebola virus causing hemorrhage and bleeding. But just as equally, the host response is at fault. I want you to remember that for the rest of this course, because I think at first glance, to many people who don't think much about viruses, they think the virus is completely at fault when it comes to disease. And that simply isn't true. It's our often over-exuberant immune response that is also causing a lot of the problem. When we study viral pathogenesis, many virologists have turned to studying the ability of viruses to cause disease in an animal because we'd like to know how it happens so that maybe we could prevent it. Here are some of the questions we ask. How does the virus enter? What does the host do in response? Where does the virus first replicate? How does the virus spread? What parts of the body are infected? Is the infection cleared rapidly, or does the virus stay around for a long time, cause a persistent infection? And how does the virus get transmitted to other hosts? These are just some of the questions that people work on when they study viral pathogenesis. Now, in order for an infection of an animal to be successful, 3 requirements. You have to have enough virus, and enough virus depends on the virus. For some viruses, maybe 10 plaque-forming units would be enough to initiate an infection if you swallowed the virus. However, for others, you maybe need 10,000 or 100,000, maybe a virus vectored by a mosquito. It really depends on the virus and where it's being received by the host. The virus has to encounter cells that are accessible. That means the virus has to be able to reach them. And of course, they have to be susceptible and permissive. These are 2 terms you know. Susceptible means the cells have receptors, and permissive, of course, means the interior of the cell can support virus replication. And finally, the virus has to encounter little resistance from the host. Antiviral defenses either are absent or overcome. We'll see throughout the rest of this course that often the virus encodes gene products that can overcome immune defenses from the beginning through the end of an infection. Let's talk first about how viruses get into a host. We're going to use humans as an example here, and there on the right is a human with the places that viruses enter. There's not really that many sites for viral infection. You have lots of mucosal surfaces. Those are the primary sites. Mucosal surfaces have a layer of cells that are living. Your respiratory tract, your gastrointestinal tract, your urogenital tract, these are all lined with living cells because materials have to be exchanged with the exterior. And therefore, those are prime sites for infection with viruses. So mucosal surfaces are one. Your skin is the largest organ in your body, but the outer layer is dead, so it's not a very good site for infection unless you have a scratch or an injury or an arthropod brings virus across the skin. The conjunctiva of the eyes, and that's really it when you look at this picture. Let's look at these in some detail. Here's the skin first. As I said, largest organ in the body, covered with a dead layer, the epidermis, the outer layer of the epidermis, stratum corneum, these are all dead cells. Virus can't replicate in dead cells, so it's a great defense against virus infection. Below it is a layer of living cells, also part of the epidermis, but it is not vascularized. Some viruses can replicate in these cells, but it's not easy for them to spread elsewhere. Below the epidermis, we have now the dermis, which is vascularized. It has blood vessels, it has lymph vessels, it has immune cells, and there viruses, if they reach that part, can spread elsewhere. Now, mosquitoes are very good at probing the skin. Here's a female mosquito taking a blood meal, and female are the only mosquitoes that take blood meals. Males do not. They simply drink sap in plants and trees. But when a female needs to make eggs, she takes a blood meal. She goes to a host, inserts her proboscis into the skin through the epidermis into the dermis, seeking a blood vessel. Eventually, the mosquito will find a blood vessel, insert the proboscis into the vessel, and take a blood meal. Now, in the process of looking for a capillary, the mosquito is ejecting saliva, and that saliva is a veritable pharmacopoeia. It contains, among other things, anesthetics, so you won't slap the mosquito away. It contains anticoagulants to keep the blood flowing. And as the mosquito is depositing the saliva into the dermis, it's also depositing viruses if there are viruses in the mosquito. Here's a link to a movie I made called How Mosquitoes Spread Viruses. Have a look. It shows a wonderful series of videos of a mosquito probing for blood vessels in mouse skin. And it's really amazing. They probe, and then bingo, they hit a blood vessel and they start pulling the blood out of it, which may or may not contain a virus. So mucosal surfaces are ripe for infection because, as I said, they're lined with living cells. And let's look at some of these in detail. They're all summarized here. Let's start with the respiratory tract. This is the most common site for virus entry. The entire tract is lined with ciliated epithelial cells. On top of that, of course, is a mucus layer, which is involved in protection as well. When you swallow, which you do often, what you are swallowing in part is mucus that's been brought up from the lower reaches of the respiratory tract into your mouth so that you can swallow it and digest it. And what happens is bacteria and viruses and other particles, when you inhale, are trapped in the mucus. There's a brush border made of cilia that beat. It's called the ciliary elevator. They beat upwards, moving the mucus to the top of the tract where you swallow it, and it goes into your stomach and it's digested. Respiratory tract samples about 8 liters of air a minute. That's your resting respiration rate. And so whatever's in the air, you are sampling, including viruses. And so this is the first defense, this layer of mucus. Below it is a layer of epithelial cells, which can be sites of virus infection. And anywhere from the upper to the middle to the lower tract can be sites of virus infections. You can see here different viruses that infect the upper and lower tract, and the different clinical names we give to infections at different sites. For example, the common cold is called rhinitis. And we have pharyngitis or laryngitis, again, inflammation at these 2 sites. Then lower down, infection of the trachea, the bronchi, the bronchioles, and infection of the alveoli, the terminal sacs, would give you pneumonia. So this is a primary site of virus infection. You can see just some of the viruses that cause infections there in the respiratory tract. The alimentary tract is another one. It's, again, always taking in materials from the external world for nutrition and hydration. We drink and we eat food. We put things in our mouth that are covered with viruses, and they're brought into our stomach and into our intestines. And of course, if these viruses replicate in our intestines, we then shed them in the feces, and they spread to others by those routes. Take a closer look at the intestinal tract. It's, of course, also lined by an epithelial cell sheet. Here's an example in the small intestine where these sheets form villi, of course, with increased surface area, and they're involved, of course, in absorption of materials that have been digested. If you take a close look at the epithelial sheet, you can see above it's lined with enterocytes. These are cells that also have microvilli at the top, and they form a barrier, of course. Interspersed among these enterocytes are what we call M cells. These are cells involved in sampling the lumen of the gut to make sure there's nothing foreign in there. So they are actively involved in moving material from the lumen to the underlying tissues by transcytosis. And on the right, you can see an electron micrograph of an M cell. And it turns out that some viruses do access the underlying tissues via M cells. Here we have some virus particles binding to the surface of M cells. They can be taken up by transcytosis and move into the areas below the M cell, into what we call the subepithelial tissues. And of course, the subepithelial tissues have blood vessels and lymph vessels, and that would allow virus to spread elsewhere. A major barrier, though, to viruses getting across this epithelial barrier is this basement membrane you can see in yellow. It lines the bottoms or the basolateral sides of the enterocytes. In the gut tract, there's also a basement membrane in the respiratory tract as well. And it prevents materials from getting through. There are also tight junctions between cells, which impede progress. But as you can see here, immune cells are very good at traversing the basement membrane. And viruses take advantage of that. And viruses also cause inflammation. That is, when they infect a cell, the immune cell responds by having immune cells come to the area. Cytokines are produced that cause, among other things, the basement membrane to loosen up. So virus inflammation, which is a response to infection, can play a role in viruses spreading from the initial epithelial sheet. Urogenital tract is another site of viral entry. It has its own protections in the form of mucus and low pH, but abrasions caused by sexual activity can allow viruses to enter the urogenital tract. Sometimes these viruses replicate locally. An example are the human papillomaviruses. A papilloma is a wart. There are many different kinds of warts that we can get caused by different papillomaviruses. We can find them on our hands, on our feet, on different parts of our body. There are genital and anogenital warts caused by papillomaviruses, and these can also cause cancer. That's why we have a vaccine against HPVs, certain HPVs. Some viruses can spread from the urogenital tract where they're initially deposited elsewhere, like HIV and herpes simplex viruses, and we'll come back to those later. The eye is another portal of entry. You blink every few seconds, and when you blink, you are washing the eyelid with fluid to remove any grit or dirt that is attached to the eye, because it, of course, is exposed to the external environment. But it turns out that viruses can infect certain parts of the eyes. For example, the conjunctival epithelial cells, the epithelium that lines the surface of the eyeball, can be infected by a variety of viruses. You can see the cornea and the corneal epithelial cells can also be infected. And one of the consequences of having viruses replicate in the eye is shown in the upper left, the subconjunctival bleed, the blood vessels below the conjunctival epithelial cells are breaking and bleeding, and you see this scary looking red appearance. Virus infections of the eye often happen when you go into a hot tub which has been improperly sanitized. It has infectious viruses in it. They get into your eye and they cause these infections. And don't forget the fetus. Fetus is another part of a pregnant mother that can be infected. And the fetus can be infected in two ways. Transplacentally, in which case the mother is infected, so the mother has acquired an infection, it has circulated within her bloodstream, and then it crosses the placenta, enters the fetus, and replicates there. Or perinatally, which means at birth. At birth, there's lots of blood involved, and if that blood has virus in it, such as HIV, it will infect the fetus. We now are aware of a number of pathogens, we call them torch pathogens, which can infect the fetus and cause problems, including congenital birth defects, and these include toxoplasma, rubella, cytomegalovirus, HIV, and other is the O in torch. And recently, the latest addition to the other is Zika virus, a virus that we know can cross the placenta and infect the fetus and cause a variety of birth defects. Our first question for today is, the outer layer of which of the following is dead but can still serve as a portal of virus entry. Respiratory tract, alimentary tract, eye, skin, or urogenital tract. Pause the video because now I'm going to give you the answer. The answer, of course, is skin. The outer layer of skin is dead, but it can still serve as a portal of entry by procedures that breach it. For example, a mosquito poking through the outer dead layer, or a scratch, or a needle stick. Those can all get through that outer layer. But if virus simply lands on the skin, if there are no breaks, it will not replicate because it's dead. Now that virus has entered the host, how does it spread elsewhere? There are two situations. First, viruses replicate at the site of entry and stay there. They remain localized. So if we had a virus infecting this respiratory epithelium, it can spread from cell to cell along the epithelium, but it doesn't get beyond that. It's contained by the tissue structure. In particular, the basement membrane makes it difficult for viruses to get into these subepithelial spaces. And the immune system can also play a role. When these cells are infected, the cells are going to send off signals that call in immune cells, and they can restrict the infection. That's one category. The other is that viruses can spread. They can spread beyond the primary site. We call that a disseminated infection. A special word, systemic, means that many organs are infected as a result. In order for that to happen, both physical and immune barriers have to be breached. In particular, the basement membrane has to be breached, and the viruses have to get around the immune response. Let's see how that can happen. Here we have a magnification of the intestinal epithelium. We have our enterocytes, we have M cells, and as I already told you, viruses can enter M cells and exit the other side. They can replicate in these cells or simply be transcytosed across them, and they'll find themselves below the M cell. There, the basement membrane isn't always intact, or in some cases, cells might be moving through the basement membrane, allowing the virus to go with it. But as I mentioned earlier, virus replication in these enterocytes, or if this were a respiratory tract, respiratory epithelium, these would cause inflammation. An immune response would ensue, and part of inflammation would be the breakdown of the basement membrane, and then the virus would be able to move across it. So whether or not the virus can cause inflammation after infection is a determinant of spread. Another important determinant of spread is whether the virus, after replicating in, say, these enterocytes, is released apically or basolaterally from the top or the bottom of the cells. These cells in the tract, the respiratory or the gastrointestinal tract, are polarized. They have apical and basolateral domains that are different, and some viruses are released from cells at one or the other sites, or some are released from both. So here's an example of this. Here on the right are 3 electron micrographs. Atop is influenza virus budding from the apical domain of respiratory epithelium. So that's typical for influenza virus. And apical release, of course, facilitates dispersal. If it's in the respiratory tract, it'll be in the air that comes back out of the tract. If it were poliovirus in the gastrointestinal tract, the virus be released into the lumen of the GI tract, where it could then spread to others in the feces. Here's in panel B, measles virus also being released at the apical domain. This is another respiratory pathogen. And on the bottom, you can see vesicular stomatitis virus, which is being released on the basolateral domain. Basolateral release gives the virus access to the underlying tissues, the subepithelial space, and eventually the entire organism, because there lie the blood and lymph vessels. The virus can get into those and spread systemically. An interesting story with Sendai virus illustrates the importance of directionality of virus release from cells. Sendai virus is a paramyxovirus of mice. Normally the virus, when given to mice by the respiratory route, will cause a respiratory infection. A mutant of Sendai virus was made, single amino acid change in the viral glycoprotein. The virus now is released both apically and basolaterally. I should say that the wild-type virus, of course, is only released apically, and it is confined to the respiratory tract. The mutant that is released both apically and basolaterally can spread systemically in the mice, killing them. So the simple fact of changing where the virus is released can affect whether the infection remains localized or spreads in the organism. Once in the subepithelial spaces, the virus then has access to the blood and lymph circulatory systems. So here we have our body surface epithelium. The virus has traversed the basement membrane. It can now enter a lymph capillary. These are quite permeable, more so than blood capillaries. It could also enter the blood, but typically the virus enters the lymph and go through a lymph node and eventually get back into the blood circulation. And there, of course, the virus is brought throughout the body and could potentially replicate everywhere. Virus in the blood is called viremia. And here's a chart that shows an experimental infection of mice. And I'll use it to illustrate the different kinds of viremia there are in a host. In this experiment, a mouse was inoculated intravenously in the tail vein with a certain amount of virus. And then at different times after infection, samples of the blood were taken from the mouse and virus titer was determined. And that's shown on the y-axis. So you can see at time 0 and shortly thereafter, you have a spike of virus, which is what the inoculum is. That's passive viremia. When you inoculate virus into a host, whether it be via a needle or a mosquito, what first ends up in the blood is passive. It hasn't replicated yet, so we call it passive viremia. In this example, 2 days later, we have what we call a primary viremia, and that is the result of the inoculated virus replicating in some cell, typically near the site of inoculation. Primary viremia simply means the virus produced by replication at initial sites, initially encountered cells, and that replication leads to virus in the blood. Those viruses can then spread further through the blood, and you can see here starting at day 4, another surge of virus production in the blood, which goes much higher than the other two. We call this secondary viremia because this is the consequence of a second set of cells that were infected. They were seeded by the primary viremia, and they're being infected and virus is being released back into the blood. So 3 distinct stages: passive, primary, and secondary viremia. And virus in the blood has a number of consequences. The most obvious one, of course, is that in the blood, the virus can spread anywhere in the organism and has access to virtually every tissue. But practically speaking, as you know, we have not figured out how to make synthetic blood yet. So we depend on blood donors to provide the blood needed for medical purposes. If virus is in the blood that we donate, it will infect the recipient. So every pint of blood has to be checked for the viruses that we consider to be a threat to human health. I say that specifically because, in fact, every pint of blood that we use does contain viruses, quite a few of them, in fact. But if we discarded every pint of blood because it were positive for viruses, we'd have no blood supply. The fact is there are many viruses in us that don't seem to do any harm, and so we make the decision to use blood containing those viruses because they don't seem to be a threat. However, other viruses, if found in blood, will cause the blood to be rejected, like, of course, HIV. And before HIV was widely detected, many people were infected with it through the blood supply. Hepatitis B virus and many other viruses as well. But the fact is that our blood supply does contain viruses, and they seem to be causing no harm. Here's a summary of the pathogenesis of an infection in mice. It's infection of mice by a virus called mousepox. This can be used as a paradigm for how a virus spreads in the host. These were experiments done many years ago by an Australian virologist by the name of Frank Fenner. These were classic experiments that established many paradigms for understanding how viruses spread. In this case, the virus is inoculated into the footpad. In the wild, this is a natural route of inoculation. Mice are walking on the forest floor or the desert floor. Their footpads have nicks in them from doing that, and if there is virus that is on the floor from another animal, it will get into the footpad. So this is how the mouse acquires this infection in the wild. So we inject the virus into the footpad. It replicates in the skin and then gets into the lymph system, as you can see here. It actually replicates in the lymph node, and then from the lymph gets into the blood, and there we have our primary viremia. So this is an example of what I meant by primary viremia. You have inoculation and local replication, in this case, in the lymph node. So now we have primary viremia, which is bringing virus to other organs, including spleen and liver. The virus replicates in those organs. The virus produced in those organs goes back into the blood. We have a secondary viremia. And in that case, the virus is brought to another site, the skin, where the virus multiplies, and it causes the characteristic rash that you can see developing in these mice at the bottom of this diagram. So primary viremia seeds the virus to different organs. Secondary viremia then seeds it to the skin. And that's where the disease is obvious from just looking at the mice, the development of this rash. At the same time, here we're looking at day 8, 9, and 10, and 11 after infection. The foot is actually swelling. That's the foot where we inoculated the virus initially. By this time, we have an immune response that is causing swelling of the foot. It's inflammation. It's part of what inflammation is, which we'll define more carefully later on. Now, we just talked about a virus in mice that reaches the skin. And by the way, that paradigm of viruses entering at some point and going elsewhere and making a skin rash is common for a lot of human viruses like measles and smallpox and chickenpox. And these are all rash diseases. These are viruses that cause skin rashes. Now, when we talk about rashes, there are a couple of different kinds. On this particular table, we have two: maculopapular, that's shown on the left here, kind of a red flat rash, and then vesicular. These are raised and look like they're full of fluid. And you can see here different viruses that cause a variety of human diseases associated with rashes. Here's measles on the lower left, which is a maculopapular rash. Smallpox is this rash in the middle. This is an extremely serious illness, which of course has been eradicated by immunization. There hasn't been any smallpox since the early 1980s in humans, but this infected humans for many, many, many years, has a 30% mortality rate, and is completely disfiguring. You never really heal from these rashes. Your skin is always marked. And then we have chickenpox, which is a vesicular rash. And when I was a child, everyone got chickenpox because there was no vaccine. Nowadays, there is a vaccine, and it prevents the infection. When we were infected as kids, it was called chickenpox for that first exposure. It's associated with this rash disease, which kept kids home from school for a week. And then 50 to 60 years later, after this, you would have a reactivation of the same virus leading to shingles. And that's because the virus stays with you from that first infection for your entire life. We'll talk about that later. So different viruses can cause rashes in humans. And the way these viruses get here, we'll talk about a little bit. These three viruses that are causing skin rashes in humans, these all entered via the respiratory tract. They traversed the basement membrane of the epithelium, they established a viremia, and eventually got to the skin. So the principles we established in mousepox help us to understand how these viruses are infecting humans. Our next question is, in general, secondary viremia is a consequence of which of the following events? A, viral replication in the bloodstream. B, viral replication at the original site of entry. C, replication in organs distal to the site of entry. D, apical release from cells. E, all of the above. I'm going to give you the answer now, so if you want to pause, now is the time to do so. The answer is secondary viremia is a consequence of viral replication in organs distal to the site of entry. Remember, the original site of entry gives you that primary viremia. The virus then spreads in the blood somewhere else, and that is secondary viremia. Besides spreading in the blood, viruses can also spread in nerve cells. This is called neural spread. And on the left is an indication or an illustration of neural spread. Here's a diagram of the spinal cord and a neuron that is innervating a muscle and also a sensory nerve ending. And viruses can enter sensory nerve endings or motor end plates in muscles. At these sites, there are junctions of nerve cells with other types of cells, muscle cell or a skin cell, for example. Viruses can enter the nerve and travel all the way to the spinal cord. For example, poliovirus can enter the nerves in muscles and be transported to the motor neuron in the spinal cord. Herpes viruses can enter via the epithelium, enter sensory nerves, and end up in the sensory neurons in the dorsal root ganglia. This movement of viruses is by the transport mechanisms of nerve cells. Here on the upper right is a diagram of that. So here's a nerve cell with the cell body on the left and a long axon or dendrite. And at the other end is the nerve terminal. And of course, when signals are passed from nerve to nerve, this is where the signals would be passed at the nerve terminus. Now, if a virus infects the terminus, this could be in a muscle or a sensory nerve ending, the virus would be taken up into a vesicle and transported along microtubules up the body of the axon. And here you can see in this inset a virus being transported on a microtubule in a vesicle. It's attached to the microtubule by a motor protein kinesin. And that virus will be brought up to the cell body, and eventually it would be released where the virus can start replicating. So we believe that virus transport in nerves occurs by these mechanisms. And we can also demonstrate anterograde transport. Viruses produced in the cell body can be brought away from the cell body and released at the nerve terminus. At the bottom is an image of neurons of an animal infected with a herpes virus that produces green fluorescent protein. And this illustrates the fact that one can use the ability of viruses to be transported in nerves to actually map the wiring of nerves in the central nervous system. So in this experiment, the virus was inoculated with a fine needle stereotactically at one point, and then sections are taken at different points after infection, and the production of green fluorescent protein can be used to track the progress of the virus across different nerve cells. And in so doing, you can actually map all the connections of the different nerves. And so you can see here, these are all synaptically connected neurons. And so the virus will move from a nerve terminus to the cell body, to another nerve terminus, to a cell body, and so forth. So this is being used in many laboratories to not only map connections, but to specifically infect certain neurons and see how they participate in different activities. When we talk about infections of the central nervous system, we use specific words. I want you to know what these are because I'll use them. First is neurotropic. Neurotropic means the virus can infect neural cells. It doesn't matter how the virus got there. It could be from the periphery, from a nerve, from a blood vessel. Doesn't matter. As long as the virus can infect and replicate in the cell, it's neurotropic. Neuroinvasive is the word we give to the ability of a virus to enter the CNS after it's inoculated at a peripheral site. So if we inject virus in the tail vein of a mouse and it gets into the brain, that's neuroinvasive. If we inject virus into a muscle and it gets into the brain, that's neuroinvasive. And finally, neurovirulent is a virus that can cause disease of nervous tissue. So if we inject poliovirus into the brain of a mouse and the mouse becomes paralyzed, that virus is destroying nerve cells. It's neurovirulent. And here's some illustrations to help you distinguish between these three definitions. First, we have herpes simplex virus. This is a virus of low neuroinvasiveness. It's not easy for it to get into the central nervous system, but if it does, and it does so rarely, it's very neurovirulent. It's highly neurovirulent. It causes disease. Mumps virus is highly neuroinvasive. In fact, back in the day when kids weren't immunized against mumps because there was no vaccine, a lot of kids got mumps, and about half of them had virus in their brains, but they were fine because the virus was not neurovirulent. So this is a virus with high neuroinvasiveness, low neurovirulence. And finally, we have rabies virus, which is highly neuroinvasive and highly neurovirulent. So this is the worst combination. You get bitten by a rabid dog, it's highly likely that the virus will go into your brain and spinal cord. And once it's there, it's likely to kill you. In fact, rabies is nearly 100% fatal if you're not immunized. It does take a while for the virus to get from a peripheral site into the CNS, especially if you're bitten on the hand. You have time to immunize. And so that's why we immunize people against rabies after the actual bite. Now let's talk about how viruses get into tissues. We've got them spreading in different ways by the blood, via nerves, how do they get out of these into tissue? So we'll talk about getting out of the blood mainly. And here are 3 different ways, going from tough to easy. And on the left is a diagram of a capillary in certain kinds of tissues that are listed below, including CNS, heart, connective tissue. Here we have a capillary which is made up of endothelial cells. You can see one nucleus there. They are joined with tight junctions, so it's very hard for anything to get across them. The blood would be in the middle there. Around the endothelial cell, around the capillary, is a basement membrane, and the combination of the 2 tight junctions in the endothelial cells, basement membrane, makes it really hard for viruses to get into the surrounding tissues from the blood. In fact, in the brain, if this were brain, we would call this the blood-brain junction. It is a barrier that prevents things from getting easily from the blood into the brain. That's to protect the brain itself. Now, the middle example is a little more easy to traverse. Here we have a capillary where the junctions between endothelial cells, here's one endothelial cell, here's a second. They're not too tight, there are pores in between them, so viruses can get across, but there's still a basement membrane, so that has to be dealt with. And as I told you, basement membranes can be dealt with by causing inflammation. So you can imagine a virus might infect this endothelial cell, and the resulting inflammation could disrupt the basement membrane, so that would be a way for viruses to get across. And finally, on the right, you have what we call a sinusoid. And this occurs, for example, in the liver. And here the blood vessel is really just a loosely formed cavity. And you can see these 3 endothelial cells are not even joined. Blood would be flowing through here. Very easy for viruses in the blood to infect the underlying cells. There's no basement membrane. And so this is why in the liver, for example, it's very easy for viruses to have access to liver tissues. Let's take a closer look at this blood-brain junction. So here on the top image is our capillary, and imagine this is in the central nervous system, the brain or spinal cord. And there, just for size, is a red blood cell and a T cell flowing through. You can see the endothelial cells that form the capillary are tightly joined. But even in addition to that and the basement membrane, around it is a astrocyte. These wrap around the capillaries and give them further protection. And you can see this junction is expanded at the bottom here to show you how tight the endothelial cells are joined. There are tight junction proteins that mediate the joining. In fact, they're quite extensive, as you can see on the right. This is a blown up view of one of these tight junctions. You can see there are multiple proteins that cross the membrane of the neighboring endothelial cells, really to bring the membranes together and prevent viruses and other things from going through readily. However, as I said, this can be breached. Sometimes viruses will infect astrocytes, and these will react and cause a loosening of the endothelial junctions, a loosening of the basement membrane as well. Here's an overview of tissue invasion in the CNS. On the left is a single capillary in the brain or spinal cord, and it summarizes the different ways that viruses can get across it. You can see here, again, the endothelial cells forming the capillary, tight junctions with a basement membrane. We have a few ways shown here of viruses getting out. First of all, viruses, the little red dots, can replicate in endothelial cells, cause inflammation, get through the basement membrane. They can be transcytosed, taken up into vesicles, and simply moved across. If they infect an immune cell, a lymphocyte or a monocyte, those cells are very good at crossing these tight junctions. This is a natural process for them. They have to get out of the blood into the surrounding tissues. For example, if there's an infection in the tissues, these cells will home to it and go through out of the capillary into the tissue. And if those cells are infected, of course, they will bring virus with them. On the right is an overview of ways to get into the brain. And we've talked about viruses getting in via nerve pathways from muscle or sensory nerves. Viruses can come through cerebral blood vessels using the mechanisms we've just talked about. There are also meningeal blood vessels that can introduce virus as well. And of course, once viruses get out of these vessels, the virus is in the brain proper where it can replicate. And there's one other place where viruses enter from the cerebrospinal fluid. The cerebrospinal fluid is made in the choroid plexus, which is a structure that is lined with cells that are much more permeable than cells in the capillaries. So viruses can get in via the CSF through the choroid plexus and spread through the brain as well. Now, lest you think that the brain is a sitting duck for virus infection, it is very well protected by these sorts of mechanisms, the astrocytes and so forth. So most viruses do not get into the brain, but some do, a handful do, and we'll talk about some of those in this course. And then once a virus is in the brain, it's trouble because the immune responses aren't as vigorous there as they can be elsewhere. And so viruses typically can replicate unchecked and cause problems. So we've talked about viruses getting through capillaries into tissues, but most viruses don't replicate in every tissue that they enter. In fact, most only replicate in a subset. And this spectrum of tissues infected by a virus is called its tropism. And we often use words that describe the specific tissues that are infected, like viruses that infect the enteric tract we call enterotropic, neurotropic for nerve cells, hepatotropic for the liver. And some viruses are strictly hepatotropic, others have a wider tropism. And in fact, some viruses can infect almost every tissue. We call that pantropic, and some viruses are limited in their tropism. And virologists are interested in understanding what determines the tropism of a virus, because if we can understand why certain tissues are resistant to infection, that may inform our ability to control virus infections. And here are some determinants of tropism that we have learned about. One, of course, is susceptibility, whether or not there's a receptor on a particular tissue. For some viruses, the receptor is very limited in its expression in different tissues, and that determines where the virus will infect. Another determinant is permissivity. The internal environment of a cell may not support virus replication. That could be due to a whole number of other factors, including, say, transcription factors, enhancer binding proteins, translation proteins, and so forth. Accessibility of cells can determine whether a virus will infect them. If a virus can't reach certain cells, it won't replicate in them. And of course, immune defenses are also a determinant. Let me give you an example of this. The poliovirus receptor, as far as we can tell, is on most tissues in the human body, but the virus only replicates in the gut and in neurons of the central nervous system. It turns out that the determinant of poliotropism is the immune response. And if you knock out immune defenses, in particular knocking out the interferon system of the mouse, the virus can now replicate everywhere. So the only thing that's keeping poliovirus out of most mouse tissues, and this experiment had to be done in mice, of course, is the presence of a vigorous innate immune response. If you take that away, the virus can replicate. I want to tell you about one determinant of tropism that was discovered for influenza virus, and that is cleavage of the HA molecule. You may remember the influenza virus particles, which are shown here, have a hemagglutinin protein on their virus surface. That protein, of course, is important for attaching to cell receptors and catalyzing fusion when the endosome pH drops. The HA protein is diagrammed at the top here. The extra virus or extracellular part is in red, and it's embedded in a membrane, of course. And I want to point your attention to the fusion peptide there at the right end in green. That is the fusion peptide that's normally buried near the membrane, near the virus membrane. But when the pH drops in a cell, the fusion peptide is exposed on the very tip of the HA and inserts into the endosome membrane, catalyzing fusion. Now, that fusion peptide is normally buried near the virus membrane, and furthermore, it has to be cleaved to generate the N-terminus that will eventually move to the top of the HA and insert into the endosome membrane. If you want a refresher on that, go back to the lecture on virus entry. That would be lecture number 5. The cleavage of this fusion peptide is a determinant of influenza virus tropism. And here that's illustrated on the bottom. Here we have influenza viruses being released from the epithelial cells lining the respiratory tract. As released, these viruses have uncleaved HA. That is, this fusion peptide is not exposed by cleavage at that orange arrowhead. However, there are cells throughout the respiratory tract called club cells that produce an enzyme, a protease or tryptase, and that tryptase is thought to cleave the HA at the N-terminus of the fusion peptide. This cleaved HA can now infect another respiratory epithelial cell. Upon lowering of pH in the endosome, the HA will fuse with the endosome and the virus infection will proceed. This enzyme, this tryptase, is thought to be limited to the respiratory tract, and that's why influenza virus tropism is limited to that site. If these influenza viruses with cleaved HA, let's say, got into the blood and went to your liver, those viruses could enter your liver, infect a liver cell, but the viruses produced by the liver cell would not have cleaved HA because the protease is not present. Therefore, the infection would not spread throughout the liver cell. So we say in this case, the tropism of influenza viruses is determined by this enzyme that cleaves the HA. Now, there are other influenza viruses that infect birds. These are called H5N1 avian influenza viruses that occasionally spread to people and cause very serious disease, which is characterized by not only replication in the respiratory tract, but apparently in other organs as well. So what's going on there? It turns out that the hemagglutinin of highly pathogenic H5N1 influenza viruses have an insertion of multiple basic amino acids at the cleavage site for the fusion peptide. And that allows cleavage to be carried out by furins, which are proteases that are present in virtually every tissue. So you're no longer limited by tryptase produced by a club cell in the respiratory tract. The H5N1 influenza viruses can spread to other organs, and they will replicate productively there because those organs contain furins that will cleave the HA and allow productive infection. So a good example of how the tropism can be changed by the insertion of amino acids at the cleavage site for the fusion peptide so that a different protease is involved. Our next question is, insertion of multiple basic amino acids at the HA cleavage site allows influenza virus to infect many organs. This means that the blank of the virus has changed. Susceptibility, club cell tryptase, permissivity, tropism, or all of the above. Or all of the above. If you would like to hold the video now, because I'm going to give you the answer is tropism. The insertion of multiple basic amino acids at the HA cleavage site of these H5N1 viruses allows the virus to infect many organs, and the tropism has changed. It's been expanded. Now, so far we've talked about viruses entering a host, spreading throughout the host, infecting different tissues. We'll look in some more detail at that part as we move on in other lectures, but today I want to talk about the other required step, and that is to get out of the organism and find another one, to find another host. And that is required to maintain the chain of infection, otherwise the virus is gone. If a virus infects one host and doesn't get out to find another, that's the end of that particular virus. So transmission of an infection is essential for maintaining the virus in the ecosystem. And in general, there are two patterns of transmission, and they're shown on this slide. We have human to human transmission. So this human on the left is infected and is shedding virus in some way, and that virus infects another human. This is probably the most common way that human virus infections are transmitted. But the other is when you have a vector that transmit the infection. And that's shown on the right. We happen to use rodents here as an example, but you could just substitute this rodent for a human and it would be the similar principle. So here we have a tick that is transmitting infection by taking a blood meal on a rodent. The rodent gets infected, and then another tick will take a blood meal from that rodent and get infected and pass the virus on to another rodent, and so forth. And so viruses can be transmitted among humans either by ticks or by mosquito vectors. When we talk about transmission, again, there are terms I want you to be able to understand horizontal transmission is between members of the same species. So human to human is horizontal. If I sneeze in your face and give you flu, that's horizontal transmission. If a mouse bites another mouse and gives it an infection, that's horizontal. If I got a virus from a chimpanzee, that would be a different species. We would call that zoonotic transmission. If you're in the hospital and a nurse infects you with a contaminated instrument, that's called an iatrogenic transmission. And it would also be, if you happen to be in the hospital when that nurse infected you, a nosocomial infection, something you acquired in a hospital or healthcare facility. Vertical transmission is between parent and offspring, mother to child. And finally, germline transmission is when the agent, the virus, is transmitted as part of the genome. So a provirus, a mouse transmitting a provirus to its offspring, the provirus then makes virus, which is infecting the offspring. Now, our human proviral DNAs do not make infectious virus. So humans, as far as we know, do not do germline transmission of infectious viruses, but they certainly do germline transmission of proviral DNAs. In order for viruses to be transmitted, virus shedding is often required, but not always, as you'll see. So if you have a respiratory virus infection, you're often shedding virus in sneezing, coughing, respiratory secretions, mucosal shedding from the respiratory tract. If you have an intestinal infection, you are shedding virus in the feces, and it can be spread to others through fecal contamination of foods. Sexual activity will also transmit virus in either urine or semen. But there are other ways that viruses can be transmitted. You could have skin lesions and rub someone and transmit the virus that way. Wrestlers often do that with herpes virus infections. You could have insect vectors taking your blood and transmitting it to another. So now we're in the category of non-shedding transmission. So if you are an intravenous drug user and you share a contaminated needle, you could get the infection that way. Infection via the blood supply, insect vectors, germline transmission. These are all non-shedding modes of transmission, right? Because you're not exhaling the virus or transmitting it in the feces, or you're not bleeding. It is taking blood from you in the form of a blood donation or an insect vector. And of course, germline is transmitting it in your DNA. So a few cases where there isn't shedding, but these are all ways that viruses can be transmitted. We have vertical transmitting here as well, which of course is during birth. Here's an example of how virus is shed when you sneeze. This is a photograph of a young lady sneezing, of course, and it's backlit so you can see all the particles that she's sneezing out. These are all particles full of liquid, of course, mucus and respiratory secretions. And these contain virus, of course. They originate in the respiratory tract. And you can see some of these particles are very big and they fall to the ground close to her, and others are small and they will travel a long distance. And that's illustrated here on the right. Here we have a couple of different kinds of droplets generated by sneezing. Large droplets that have a range of 1 to 3 feet, smaller ones which go a little farther, and then very, very small ones called infectious droplet nuclei. They can go up to 160 feet. And it turns out that these droplets, in fact, the smaller ones, are also made by speaking and breathing. So you don't have to sneeze to make those small ones. The larger ones, of course, if someone is standing near you, you could infect them or very close. But the very small ones are insidious because they can be generated by breathing or speaking and travel long distances. So you could have a respiratory infection and not have symptoms and be shedding lots of virus and infecting other people. We can also spread respiratory viruses by contamination, nasal secretions on your hands, tissues, desks, doorknobs, and so forth. Nice video here, slow motion sneezing of people. Yeah, sneezing in slow motion. It's pretty graphic if you want to check it out. Recently, we talked about a study using this machine called the Gesundheit 2. And here it is on the left. It is a machine designed to sample your breathing and sneezing and coughing. And what was done in this study, they took 156 individuals in a college community with confirmed influenza. They had them sit in front of the Gesundheit 2 several times, different time points. What you do is you put your head in here, and there is an intake of air at something like 120 liters a minute. So there's a whoosh of air coming in. And they have these individuals talk, they recite the alphabet. And that's where we learn that just by talking, you are excreting fine aerosols that contain virus. Even breathing does that as well. And if you sneeze, and a lot of these people did because they had flu, it doesn't make a lot of contribution to virus shedding. They could find virus in cough, but that wasn't necessary for infectious aerosol generation. It's really just breathing and speaking which generates a lot of the transmission, which is not to say that you shouldn't cover yourself when you sneeze or cough. But in fact, talking and breathing is probably the main way these infections are transmitted. So it's an insidious means of transmission, which means that you don't even know you're doing it. As I said earlier, some virus infections lead to the formation of rashes on the skin, and that can transmit infection. And I wanted to show you these two cases. These were reported in Morbidity and Mortality Weekly Report, which is published by the Centers for Disease Control and Prevention. And it's free. You can go online and see all these. And these are transmission of vaccinia virus after sexual contact. Vaccinia virus is the vaccine that we use to prevent smallpox. And as I said earlier, smallpox was eradicated some time ago. So we no longer immunize the general population. However, we do immunize military personnel. In case of a bioterrorist attack, they would be protected and could engage in combat. So what happens is they went to get a smallpox vaccination. They take some vaccinia virus vaccine, and it is scraped into your outer layer of skin with a needle. We'll show a picture of that later when we talk about vaccines. But within a week or so, you have the development of a blister on the skin surface, which eventually crusts up. And this contains infectious virus. So when you receive this vaccine, you are told to not engage in close contact with anyone. And in fact, to keep the Band-Aid on the site of immunization for some time. But in these two cases, the individuals did not heed the advice. They had sexual contact shortly after immunization, and they spread the virus infection to their partners. Now, I will tell you that the virus, when it forms a lesion on the skin, is quite bearable, but this virus can also infect mucosal surfaces, the kind that are exposed during sexual activities. And there the infection is much more painful and not something that you want to have. I'm sure the recipients of this vaccinia virus infection were not too happy. Which statement about viral transmission is not correct? All virus infections are transmitted by shedding. The route is determined by the site of virus shedding. Transmission is required to maintain a chain of infection. Speaking can produce an aerosol that can transmit infection. Horizontal transmission is among members of one species, which is wrong. You can pause now because I'm going to tell you the answer. The statement that is not correct is all infections are transmitted by shedding. Shedding is when virus goes away from your body, like in sneezing or in fecal transmission. There are many ways that we spread infections that are not by shedding. And one example would be via a mosquito picking up your infected blood. Before we wrap up today's discussion, I want to touch on 2 points that are relevant to virus infection. And that is that geography and season can affect when virus infections are transmitted. And let's look first at geography. a given virus infection isn't always all over the planet. Some are geographically restricted. And that is sometimes, not always, but sometimes because there is a source of the virus or a vector that is geographically limited. So let's, as an example, and there are tons of examples of this, and you'll recognize them as we go on in this course. Let's look at a virus called chikungunya, and in particular how the vector can affect the localization of infection with this virus. Chikungunya virus is a toga virus. We've mentioned these briefly before. They are enveloped plus strand RNA viruses. You may remember them because these viruses in their replication scheme make a subgenomic mRNA. These viruses are vectored by Aedes aegypti mosquitoes. When you get infected with this virus delivered by the bite of an Aedes aegypti, you develop rash, fever, and joint pains. Now, where is this virus infection seen? Historically, it was mainly in Asia and Africa, and that's shown by the blue on this map. But around 2004, there was an outbreak first in Kenya. It spread to India, and then an outbreak in this small French island of Réunion. And Réunion is a popular visiting site for tourists from Europe. The virus then spread back to Europe, Italy, and then throughout Europe itself. So starting in 2004, the virus went outside of its normal range. And now we have these orange sites of infection here. So what happened in 2004? Well, what happened was a new vector became able to transmit infection. And the new vector is a mosquito, a different mosquito called Aedes albopictus, also known as the Asian tiger mosquito, shown here. Previously, the virus could replicate in this mosquito, but it didn't replicate well enough to be transmitted. The way virus transmission typically occurs in mosquitoes, the mosquito takes a blood meal, virus goes into the mosquito gut, replicates, spreads throughout the mosquito, makes its way back to the salivary glands, and then when the mosquito takes another blood meal, it's ejected with the saliva. And so this virus never did very well in Aedes albopictus, but after 2004, the virus sustained a 1 amino acid change in the viral glycoprotein. That's that glycoprotein on the surface of the particle that's important for virus attachment to cells and fusion. 1 amino acid change was enough to allow high enough replication in Aedes albopictus so that it was now a good vector. We call it competent vector for transmission. And Aedes albopictus is far more globally distributed than Aedes aegypti. And you can see we've had some imported cases in the US. These are not locally transmitted cases. We do have Aedes albopictus in quite a few places in the US. Its range is shown in this map on the right here in yellow. So those are all the potential areas where the virus could spread. On the left, this map with the blue states, these are all states where cases were imported from other countries. And again, as I say, there hasn't been any locally acquired chikungunya in the US yet. But this mosquito being here, it's possible. And these are the number of cases imported into the US in 2017. But its global range has expanded because now it is being transmitted by a different mosquito that has a different range. So an example of how geography can infect the range of a mosquito, in this case, limited by Aedes aegypti and expanded by Aedes albopictus. By the way, at the bottom here is a chart predicting the increased temperatures in Europe starting before 2011 and going into the future. And you can see the warm climates moving farther and farther north. And as this happens, the range of these mosquitoes will increase as well. And that will increase the area where this infection is transmitted. And this is just one virus. Think of all the other mosquito-borne viruses whose range will increase globally as the planet warms and the mosquito range increases as well. The last issue I want to touch on, and very briefly, is that many virus infections are seasonal. They happen at specific times of the year. And we have 3 illustrations of this here. The top left, rubella, from 1963 to 1968. Here on the y-axis, the number of reported cases, and on the x-axis, these are yearly cases reported. You can see January to January 63, 64, 65, etc. And you can see a distinct peak in the number of cases here about May every year. On the bottom is influenza, which is a winter disease. You can see peaking in the winter months, March, January, every year. Even poliomyelitis, when it was very common, had seasonality depending on the latitude. Up in Alaska, you can see it peaked in September, and down in Australia, it peaked in March. And polio is typically considered to be an infection that is transmitted in summer months. Why this is the case, why certain virus infections show seasonality, is generally obscure. There has been some work done on influenza virus which suggests that at least in temperate climates, such as here in the northeastern United States, the low temperatures and low humidity facilitate transmission of the virus in very, very small droplets generated by aerosols. The idea is that under conditions of low humidity, the droplets can travel farther, and if the humidity is higher, the droplets that you produce by talking absorb water, they fall to the ground close to you, and they don't transmit the infection as effectively. There have been some very convincing experiments to show that for influenza virus. The problem is that we do observe influenza in the tropics, and there the humidity is pretty high, and in fact it's even seasonal in the tropics. This issue of seasonality of infections, what causes it, still remains pretty much an obscure mystery. Next time we're going to start considering the barriers that viruses face when they enter a new host. And we're going to start with what we call intrinsic and innate defenses.